Dr. Kleitman is a neuroscientist who originally trained in Illinois and was then recruited to spend a number of years at the Miami Project. And Dr. Kleitman is really a pioneer in spinal cord injury and the neuroscience associated with spinal cord injury. And about 7 or 8 years ago, around 2000, she was recruited to the NIH. And Naomi is now director of the National Institutes of Neurodevelopment and Neurologic Disorders and Stroke. And she's going to kind of set the stage this morning for some of the history of the field of spinal cord injury and what she sees are some of the barriers to cure. Naomi?
Thank you very much. I'm sure that was a wonderful introduction. I didn't hear a word of it because we were busy playing with it. But it is a pleasure to be here and see both the Shepherd Center and at least my grantees and applicants from the university as well. So I was asked to give a basic introduction. I actually prepared I think a lot about translation of findings from the laboratory to the clinic. And so I sent Anne some slides on some of my translational talks. She said, well, let's back up a little bit. So what I'll do today is first to set the scene for those of you who are not as familiar with spinal cord injury as some of the people in the audience. And mostly the rest of the time I will tell you nothing. I will ask questions. And I think that that's actually what we're supposed to be doing today. And let's see if there's... Okay, Steve. Maybe it's the wrong... I'm pressing the wrong one. Good start. Okay. So what is translation? Translation is really the missing link. Everyone knows that... so I'm from NIH. My program is in spinal cord injury research, peripheral nerve injury research. I'm in the repair and plasticity group, so obviously we are interested in the basic science of defining these types of injuries, but more interested in what we can do about them. So from the time I got to NIH in 2001, my focus has been on the translational questions. And traditionally, NIH funds basic science very well. People don't always realize it, but we do very much want to and fund clinical science. And in the long run, this has been, the link between these two has been what hasn't worked so well. The process of applying new ideas and insights and discoveries generated from those basic science labs to treatment or prevention is what we're really here to talk about. So I thought about that question. I thought, okay, well, we use these words in science, and they have real meanings in the real world. So what does translation really mean? To translate means to change the form of something, its nature, to actually transform it into something it wasn't, or to take one thing and carry it from one position to another. This is a standard, some sort of standard English. What it doesn't mean is I'm a researcher, and I have a line of scientific inquiry, and I want you to fund it, and I will write at the bottom of my abstract that my ultimate goal is to cure your disease. And tell me which disease you're interested in this year, because I can make my work fit that. It also doesn't mean I'm a clinician and I want to do a trial and somebody wrote a paper about Schwann cells, and so I'm going to take Schwann cells and do something with them. Now, I'm going to pick on Schwann cells because that's the area that I did research in for many years, but it can apply to your favorite therapy of choice. And unfortunately, I think you see a lot of these kinds of interpretations when you look between the lines, and so let's talk about how to make these two work a little bit better together. So what's our challenge in spinal cord injury? We have a very big challenge. This is a human spinal cord. This is actually a slide courtesy of Bruce Dobkin at UCLA. This is a rat brain and spinal cord. And we've got a lot of issues in terms of moving from the rats to the human. And this gets at one of the first points, which is rats are a reasonably good model with which to do some of this basic science. And the question is, what really does that mean about human injuries? So the first thing you see is pretty obvious, is this cord is that long and this cord is that long. The cervical area is about here. This is the cervical enlargement in the human cord. And that length issue is a big deal when what you're talking about is regeneration. where you're talking about 100 times different scale. The size of the lesions are also of a different scale. So here you may have a gap of a couple of hundred microns, whereas here you may have a gap in the lesion area. Because remember, for those of you who are not spinal cord researchers and don't think about this all the time, what happens in a spinal cord injury is a particular area is injured. The connections that reach beyond that in either direction ultimately die back because of degeneration. But the actual hole in the cord is a relatively limited area. So you've got a difference in the size of the hole, also the position of the hole. Much animal modeling, and this is largely because regeneration was the goal of generating the models, is done in the thoracic area of the cord, which is where there's a lot of pathways going through, but not a lot of gray matter neurons that really drive motor neurons in that area. So you can really study long tract regeneration. Whereas a lot of spinal cord injuries in humans happen in cervical cord or in lower thoracic than is usually tested in animals. But that's okay because it depends on the question you're asking. The actual tracts themselves are different. The biggest difference really in terms of control of motion in rats and primates, I'll say, not just humans, is the organization and position of the corticospinal tract, the tract that reaches straight from the cortex in humans straight to the motor neurons in the spinal cord that ultimately drive the muscles to move. In rats, for instance, it's in a different part of the spinal cord. It's deep in the middle of the spinal cord as opposed to out on the sides in the human. But also those projections from the brain in rats and in a lot of other non-primate species actually have to go through several connections before they touch the motor neurons. There's not the same kinds of direct connections as in the primate. And the last thing is the actual functions that we're talking about. So a rat walks on 4 legs. We do locomotion. We talk about whether its tail is up. We're not going to be so concerned about that in our patients. So there are different functions that we have to talk about in terms of our clinical trials than what we're actually modeling and recording in our proof of concepts in our animal models. So it's a few problems. So we have another challenge in spinal cord injury. One of the things that we do with our animal models is we make them as reliable and repeatable as possible. We want to be able to repeat from experiment to experiment, from group to group to do a comparison. Not only that, we want to be able to repeat our work from lab to lab, okay? Human injuries vary widely. This is spinal cord this is the cord. This is the muscles around it. This is what should be a nice open space for CSF to flow through. This is a human spinal cord injury which generated this lesion. For those, I am going to do basic 101. So please, I'm sorry if I'm speaking things you already know, but I'm a basic scientist, so I put the dorsal part of the cord on top. So this would normally be where sensory information would come into an intact cord. This is where the motor neurons would be sitting and sending their axons down out of the spinal cord. In this case, red means myelinated tracts, so these are the long tracts traversing that area of cord. The gray matter should have been in the middle. There's a little bit left here. On this side, it's completely smashed. And this is the type of cavitation which is seen very, very commonly in most human injuries. This was an incomplete injury because one side was much more intact than the other. This is a very, very severe spinal cord injury. In this case, the myelinated tracts are the bluish, or they're showing up sort of grayish tracts. This person had no function whatsoever going through that area, and yet there are a rim of myelinated axons, and I think you'll hear a lot more from some of our later speakers about the possibility of that. But Weiss's work was seminal back. He and Andy Blight published a lot of work on the cat models determining how much loss of function there is, how much possibility there is for recouping some of that function with the axons that do remain. But this is why, for those of you who really only know about spinal cord injury, when it shows up in the papers, they say, oh, well, it's okay, the cord wasn't severed. And we say, well, the cord is almost never severed, but that doesn't mean that the tracts themselves aren't pretty much blown away. So the functional levels, this is something else that's very important to understand. The point of that last slide was the variability. So when you're going into a human trial, you're dealing with humans with all kinds of injuries. And how do you stratify that? How do you aim your therapy? Well, one of the big issues is, does the spinal cord injury happen in the neck? Very rarely happens in the middle of the thoracic. These vertebrae have these things called ribs and a whole bunch of organs that kind of hold that pretty well. So you get a lot of injuries down here in the lower thoracic area, lumbar, motor vehicle accidents, high velocity types of accidents, or in the neck again, where all types of things can cause the injury. So a person with an injury in the neck, tetraplegia is the medical term. People may also be familiar with the term quadriplegia. The alternative is paraplegia, so I'll just explain why the change. So it used to be quadriplegia and paraplegia, but one was Latin and one was Greek, and so they said, if you're going to use one, you should be consistent. So I guess about 10 or 15 years ago, the clinical community started using tetra to go with para. If you have a very high cervical injury, you have to worry about the nerves that are involved with breathing, and not only that, but the circuitry within the spinal cord, which is the computer which generates the breathing rhythms, which is in the gray matter of that area. So here we're talking about a real issue with the hardware of the spinal cord. Below that, a lot of these functions we're talking about are signals that make it down the spinal cord to a certain point, but the injury is there, and so everything below it is gone. So when you're talking about a high injury, you have this plus everything else, and the same thing as you go down the cord. You go down shoulder to elbow, mainly proximal to distal, first in the arms, then in the legs, and don't forget, it's not about only motor, it's about sensory, coming back the other side, it's about autonomic functions. Almost everyone with a reasonably severe spinal cord injury has problems with their bowel and bladder control. And if you don't take care of those things and get a urinary tract infection or a kidney infection, you die. So these are very, very significant issues. The same thing with autonomic dysreflexia, where enough innervation comes from the top of the sympathetic nervous system to turn everything on, but the control below that to moderate your heart rate, your cardiovascular system is not there, and you generate positive feedback loops and you die. These are really, really significant questions that I think until the last 5 or 6 years didn't get a lot of attention from the basic scientists, but it's getting more now. When you actually ask a group of people with spinal cord injuries what they want you to concentrate on, Kim Anderson, who herself is a tetraplegic, works at the Reeve Irvine Institute at UC Irvine, ran and published, I think, a very important paper in 2004 based on an internet survey asking people, what do you think the priorities in research should be? And what she found is that, we use the term quadriplegic, this is actually a figure from her paper, they are very concerned with hand and arm function, way above everything else. Paraplegics or persons with paraplegia, their hands are okay. And as long as they don't ream their shoulders out pushing their wheelchairs around or hurt their hands for some other reason, hand function is generally not as big of an issue. Sexual function is the next, again, another one of the autonomic motor sensory functions that has to integrate, whereas for the paras, that was their first priority. This is very important to a person's identity, their feeling of worth of themselves and within the world. And quite honestly, the number of sexual function researchers in the spinal cord field is still quite low. Bowel and bladder comes next, whereas trunk stability is another issue for our tetras. That's less of an issue for most paraplegics. Again, it depends on the level of the injury, but if you don't have trunk stability, you're going to have a lot of trouble sitting through all the talks today in these chairs. Walking, movement, pain, normal sensation are issues for both groups. And there are others, but these are the ones that broke down in the survey. So this is kind of hard on my neck. So I'm going to stop there with the basic background, and I hope that's at least gotten us all to a place where we can really talk about what we're going to hear later on in the day. And especially hope that the clinicians will correct me on anything and everything I've gotten wrong, because I am really a basic scientist masquerading. What is our therapeutic strategy? So I've just described an extremely complicated neurological disorder. And what we find in the spinal cord injury field is a lot of the therapeutics that people are talking about are pretty sophisticated at this point, and the targets are equally complicated, and the strategies are sometimes even more complicated. But let's go back to the basic science, which is where you have to start with the translational question. Basic scientists look at mechanism, and especially NIH traditionally says, okay, that's fine, but what's the mechanism? You're going to throw this in, what's the drug going to do, what's the cell going to do? And the question I pose today is how much mechanism really matters. I don't think there's good agreement on what we really need to show in the lab, and I'll go into this a lot more when I talk about modeling, and what it really takes to develop a workable therapy. And that's one of the issues we have to deal with. We have no good gold standard, and I'm going to aim most of this talk at cellular therapies, although it's only a subset of what's going on in spinal cord injury and should be going on in spinal cord injury. We need to have a lot of different lines of research going at the same time. But, you know, if we had a great therapy that had gone from an animal model and is in standard therapeutic use, we would have a gold standard. We'd especially be able to see which models didn't predict that outcome. We'd especially be able to say which outcomes in our trials led us to test that. But we don't have that. We have very little consensus overall on what the adequate proof of principle is, or what the product issues should be with the cells that we might be putting in, and how adequate that animal efficacy data is to being able to translate and say, if I do this in a rat, a person should get a benefit. Or if I do this in a rat and see no risk, I can say for sure I won't hurt a person by doing this, because by definition most cell therapies are invasive just because a live cell, even if you can put it in through the bloodstream, you're still putting in a live cell, which is essentially a permanent therapy unless you can prove otherwise. So how much recovery? So that, you know, if you need to have consensus on what your adequate proof of principle is, we have to answer the question, how much recovery is sufficient to go forward? And if we're talking here about what the barriers are, I'd say one of my first and biggest barriers is the community has reached, as far as I can tell, absolutely no consensus on how much preclinical efficacy is necessary or advisable, because pushing drugs or cells to clinical use, I'll actually read this because I think it's important. Pushing drugs or cells to clinical use without adequate trial design simply wastes rather than saves time. And actually, when I first drew this slide up, it stopped there. But to be perfectly honest, striving for perfection in an imperfect animal model may only delay important clinical testing. And finding the right balance between those two should essentially be the goal of this type of conference, because there will never be 100% agreement, but there should certainly be better agreement than we have today. This is not specifically and only important to spinal cord injury research. It's huge in stroke research. It's huge in brain injury research. When I first started at 2001 at NIH, I joined a group which we had this massive upheaval in NINDS before I got there. So we went from having divisions and branches and things. There used to be a stroke and trauma division. And then we had these clusters because Jerry Fishback became the head of NINDS, and he did neuromuscular junction channel clustering. So it became clusters. Not many people know that's the reason. And stroke got broken into one cluster and trauma got broken into another, but they're all still very related. And in many ways, stroke and brain injury are further ahead in terms of answering some of this question, because they went out and did a whole bunch of clinical trials with very little basic science background, while spinal cord injury at the same time was diving deeper into the mechanisms in basic science. So both stroke and brain injury have done major revisiting of their own fields and said, these are all the things we did wrong in clinical trials. And I'll go through what some of those were, whereas spinal cord injury was trying to build up, I think at that point, the perfect therapy from the bottom up without bringing a lot of things to trial. So I think we have to learn from both literatures. So what are the therapies that we're talking about? Spinal cord injury is in many ways a huge opportunity, because testing a therapeutic strategy for spinal cord injury gives you lots of possible targets. This is actually a figure, it's not showing up perfectly well here, but I just have to say that I used to, as a scientific liaison for a group called the Miami Project to Cure Paralysis, I had to speak to people at every level, but most importantly, families and patients who were dealing with spinal cord injury for the first time, and now somebody's talking to them about nerve cells and neurons and dendrites and channels, and they're going, what? So I was so frustrated trying to talk about regeneration. I sat down one morning and drew a pencil drawing of what the different targets are, and ultimately medical artists turned it into this. So this is a very cellular look at what the issues are, with a hole in the middle being the area of that lesion. And you have, remember, cells from the brain coming down, maybe even passing by this area. You have neurons in the gray matter of the spinal cord itself, but what happens? First area of injury in that hole, you have the death of cells. They and the connections they would make below the level of injury go away. Second, you have those nerve cells that were safely outside the region of the lesion that projected, these are axons, and projected the outflow axons to the area of injury, at which point they were broken. And the process of Wallerian degeneration means that the rest of the connection died back over the next few weeks. But basically the communication was lost immediately. So again, this cell lost the input to tell it what to do. And the third possibility is the nerve cell survived, maybe right at the edge of the injury. It stayed connected to its target, but it's lost its insulation or myelin, which allows it to faithfully send the messages at the speed that needs to happen. So your therapeutic strategies are, first of all, to protect these cells from dying. The fewer cells and pathways that we lose, the better we are to start out with. The second is to go back and replace lost cells, which I think at the very beginning was where stem cell therapy was thought to be aimed. The third, of course, and this was my area, was to promote regeneration of these nerve fibers, because they do go away forever, unless there are some people looking at resealing and preventing those from dying, but I think we're pretty far from that. And the first, there's really two sides to this, and thanks to Martin Schwab, we know that not only do you have to drive these nerve cells to grow, but you have to block the things within the area of spinal cord which will prevent them from growing, and that's removing inhibition. You could remyelinate these spared axons, or you could just take what nature left you, and as we saw, those of us who saw the work at Shepherd yesterday, rehabilitate and retrain the heck out of those, relying on the natural plasticity, which does exist in the spinal cord, to train those remaining circuits. And I think we don't do enough of that. But no matter what we do, we have to combine that with whatever therapies we're going to generate. There are lots of good targets. We have cells that do lots of things. Have you actually determined which one of these targets your cells are going to be affecting? Because one of the things I'm going to talk about that we do is we do a lot of work with cells and dishes and say, look, when I put my neurons on these cells, I can grow axons, and here's a graph with 25% more axonal growth, and it's really great. And I throw it in a spinal cord and hope it does something good. And realistically, it's probably doing more to protect the surrounding area or dying and allowing the immune system to do whatever it can do than it really is doing regenerating axons. And one of the things that I think a lot of papers that we've seen have really jumped that question. So again, I'm going back here to a little bit of basic background. Apologies to those who created these models, if not have used them for most of their scientific careers. But we do have a number of very well-established models that we use in basic spinal cord injury research, and now I'm referring largely to rat research, although more and more of this is also being done in mice. And this is drawn up for, at the time I was trying to display how olfactory and sheathing glial cells had been implanted. But in this case, we've got, this is meant to be the spinal cord, the gray matter in the middle, the white matter around the sides, and a dorsal root coming into the spinal cord. And one of the models is actually to cut the dorsal root and try to get sensory axons growing into the spinal cord, into the CNS environment. Not, frankly, spinal cord injury, but certainly important to a lot of spinal cord injuries. The next, this is meant to actually display a lesion made by Jeff Raisman, which was very specifically aimed at one side of the corticospinal tract in rats, which, as I said before, the corticospinal tract in rats sits right here in the middle of the cord. In humans, it's more outside here on the edges. But because of that, you can actually try to target a single tract to ask a very elegant and precise question about regeneration in this model. The model we were using in Miami at the time was about as bad as you could get, and we were doing that quite intentionally. I'll jump down. This is meant to be either a transection or a hemisection lesion, a clean cut. Again, a model that's used very often in regeneration research because at least you know what you've got. With a full transection, you really know what you've got. You want to make it worse, make a full transection and take a chunk of tissue out. We were doing that because we were trying to build bridges of supporting glial cells in the interim. This is meant to model the contusion or concussion injuries. The meniscus injury that Weiss and a whole network of scientists created in the 1980s is one of the first and best characterized of these. There are now Infinite Horizons, the OSU device, all have different components. And at Louisville, they not only study the bang, the contusion of the injury, but also the compression and the continuing compression that helps create. This is meant to be a cavity, but these are meant to be little cells. And so you've got a great place to put cells if you build a hole. And that's one good thing. And the last is meant to be an injury that Hans has used quite extensively, which is to demyelinate but leave those axons intact. And you can do that by putting in a chemical that will kill off the myelin, irradiating it so that the area won't remyelinate, and then you can put your cells on axons which are just waiting to be remyelinated. The issues that continue to plague us have to do with the repeatability and reliabilities, moving from species to species. For one thing, when you do this injury in a rat, you get a hole. When you do it in a mouse, well, it depends on the strain, but you may get no hole at all. Is this hole in a rat the same as the hole in the human? We don't know. What functions you're going to target? Most of these injuries are made in the mid-thoracic region, as I mentioned before. And you're not going to target the same functional return in the thoracic region as you would if you made the injury higher or lower. So what is the real clinical relevance? When the meniscus model became available, everyone started to say, well, the cuts aren't clinically relevant. That doesn't happen in a human being. What does happen in a human being that's had a knife through their cord or a bullet through parts of their cord? Well, not a bullet. A bullet won't give you a clean cut. It'll give you the cut and then the compression and the pressure wound and the blast. So none of these are actually how a spinal cord injury happens in a human being. But they're the best we have, and they're good models, and they can be used, but the relevance always has to be, I think the relevance should always be stated as a model. And in this case, we don't have a good prediction of the human. So what are the targets for repair and what's in the drug pipeline? I'm not going to give you an exhaustive list. It does exist. But there are the neuroprotective strategies inhibiting the specific There's two ways to go about this. The molecular biologists and the molecularists will say, go about it in terms of, tell me exactly what the mechanism of your drug does. Don't tell me it's just a glutamate or an excitotoxic. Tell me which receptor 1, receptor 2, and I have some grants drilling down extremely deeply into the mechanism. That's one way to do good research. Or you can say, well, we have these processes, we have massive cell deaths, and we have secondary injury. Let's go with inflammation. It's going to start and stop a lot of processes at once. Hypothermia would be a good example of this. Even methylprednisolone has multiple posited mechanisms of action. And then you run into the situation where some inflammation is good, some inflammation is bad. How do we target? What's the timing? These are all really important questions. The neurorepair strategies that are out there, as I said, are either about turning a cell on to regenerate or turning off the things that block it. We know myelin is full of inhibitors, but the good thing is the receptors for a lot of those inhibitors are all the same. Maybe yes, maybe no. There are drugs that are have been tested, in fact, are in testing right now. And then we have to talk not only about cells and drugs, but there's a classic drug development pathway that, in fact, most basic scientists who haven't dealt with pharma may not really understand the depth of this process. But you go from basic to translational to clinical through the target ID and assay, which in most drug studies is not even done in animals. Animal studies are here under proof of concept. This is when you have a big drug library and you're going to throw it against one of those very well identified molecular processes to screen out and get some hits to bring through this area, which is proving concept in an animal, then saying, especially with a drug like, it's being tested in Canada. I'll think of it later. But some of these drugs that have multiple effects, you may want to take out some of the effects and add back others. Roloprem is good because it actually creates a lot of nausea. So you would want a better drug than Roloprem to do the good things Roloprem does, but to weed out the bad things it does, preclinical safety and then clinical trials. I'll get back to that. So I did I just did this yesterday. I did a PubMed search on stem cells and spinal cord injury. Quick, clean, dirty. 425 hits, 100 of those are reviews. So for every piece of basic science that's published, 4 papers, and then it's time for a review. They are every conceivable definition of a spinal cord, or of a stem cell or precursor, but this was a search I did. Every conceivable source, every conceivable stage of cell. Diverse delivery routes, often used in combinations, especially in spinal cord injury these days. Targeting, so remember I said you put the cell in, you do your preclinical and in vitro, you say it regenerates. They target regeneration, replacement, building and combining with scaffolds, myelination, neuroprotection, immune responses, delivery systems for gene therapies, you name it. You do the math and you divide these into 425 and you still get about 10 papers on any given subject. So it's not a lot of experience. These are cells, especially when you get into replacement, replacing what? Replacing nerve cells themselves, replacing oligodendrocytes to make myelin, replacing astrocytes because some people think astrocytes block regeneration. Some people, like Stephen Davies, think that in the right form, astrocytes will promote regeneration, or, and this blows my mind, to create Schwann cells, which we actually can generate billions of from a person. That's some of the work I did in Miami. But some people think stem cells are the better source for almost anything you need to generate. And overall, I go back and I'll make this point about 100 times this morning, we have no clear idea of how they work, if they work. And that really is the state of the art, in my opinion. And yet, they are in clinical use in many places across the world. They being, I'm not sure what, I think all of these caveats apply. So let's talk about one example. Doug Kerr couldn't be here, and I don't know if he even knows I have the slide. It's actually pilfered from a drawing that Paul Reier did that I added and changed a bit, but I do have his permission. But in 2006, Doug, who mostly looks at ALS, the loss of motor neurons, for many years, I remember when I first started at Miami, we were some of the work in Scott's lab with nerve cells, or transformed cells that become nerve cells, were actually generating some new motor neurons. And Gerda Verbova had been trying for years using fetal grafts to get a nerve cell, a motor neuron that was lost, to grow out to a muscle. Well, Doug finally did it in 2006. How? What a combination. He used ES cells that he pre-differentiated using retinoic acid to generate a population of motor neurons, or motor neuron destined cells. He implanted them in the cord and stimulated them to grow and used some of the mechanisms to block inhibitors. And there would not be a dotted line here. There would actually be essentially an axon-free peripheral nerve that he's asking these to grow through. And so he treated the cells here with one of these and then treated the nerve itself with the other, and then put another group of cells, Evan Snyder's C17-2 cells that he was just using as a delivery device for glial-derived neurotrophic factor, that he put down here to spice up the target, the come hither signals. And the result was he finally got the motor neurons here, the grafted cells, to turn into motor neurons, survive, grow out through the path, make a neuromuscular junction, and physiologically show that he could drive at least a little bit of function in that motor neuron. And I was asked at the time and quoted in the papers, I think this was a phenomenal piece of work. And he did it very carefully, showing that if you take any one of these components away, it doesn't work. And not only that, it stops at the point you would expect based on the mechanisms by which these were thought to work. All the components were required. Talk about a complex therapy. I actually made this slide up originally I was talking to an organization of rare diseases, all families of people with organizations pushing for one disease or another, and it was meant to talk about the promise and false hope of stem cells. These are complex strategies and long-term goals. These are not miracle cures and shouldn't be treated as such. So when you're planning for a clinical trial, let's look at some examples of this difficult problem we face. You have a heterogeneous clinical situation. I've already gone over most of this. I haven't talked much about timing, but you've got the acute, the subacute, and the chronic, also not terribly well defined, and you may have multi-trauma issues. You have the accuracy and sensitivity of the clinical assessments, which I won't go into here, but the American Society for American Spinal Injury Association, I apologize, is working very closely with another group of clinicians and basic scientists funded by a group called the ICCP, which I won't go into, are working on outcome measures that currently exist and how good they are for clinical trials and publishing papers on those. They've just generated a new autonomic standards for clinical care as well as research. And then we get back to the fact that there are no real treatment standards qualified within the surgical and other communities for a lot of these spinal cord injuries. Then you get into the trial design itself, not just the clinical situation. What is your goal for repair? I've gone over that. If you can get a statistically significant change in a rat with a thoracic injury, is that enough? This question of proof of concept. When do you actually start a regenerative therapy? So there was an example at the Procord trial where the use of activated macrophages, the preclinical data was done looking at a regenerative response both in the optic nerve and the spinal cord. And then when they went to do the trial, they moved very early, and it really became a neuroprotective trial. Same thing happened with most of the brain injury and stroke models. They weren't always regenerative, but they were very often tested at a different time than the preclinical. How do we conduct a combination therapy? All of these are new drugs or new cells. That's going to be very complicated. And when we do it, we'll probably have to do multi-center trials because the number of spinal cord injuries any given center may have that meet the criteria for the trial are probably not going to allow, at least in the higher phase trials. And those are all going to be complicated. Spinal cord community is addressing some of this. This is the group I just mentioned. This stands for International Campaign for Cures for Paralysis. It's a group of spinal cord funding agencies, the Christopher and Dana Reeve Foundation, Paralyzed Vets of America, Spinal Research in England. There are two groups from Australia, one from Japan, one in France. The Miami Project's involved. Convened a series of meetings following one that I held at NINDS in 2003, and many of the people here were at that meeting. We've generated a series, I've worked with them since, but we've generated a series of recommendations following again on this. ASNTR is another society, deals a lot with Parkinson's disease, and they looked at this question of cellular therapies and necessary amounts of evidence. And so starting with that as a basis, we published one paper in 2005 and followed up with a series of international meetings talking about what human clinical trials might look like. And these were published last year in Spinal Cord. They're freely available. The journal's making them continually available for free. So for those who haven't seen it, we went back and recalculated the control group data from a number of clinical trials and things like the MASCUS data, or the Model Center databases, to try and get at an idea of what the real rate of spontaneous recovery is, at least at the time those trials were done, and what the statistical power might be needed for a trial, what outcome measures you might use, inclusion, exclusion, and ethical issues, and then ultimately one on trial design. I was talking to a number of people about this yesterday, but out of that we created a lay publication aimed at you and all your patients, those who are clinicians, to help with your patients. It's at this URL, which I can give people, but it's at many URLs because it's freely available on the net. It's been translated into about 4 or 5 languages at this point, and it includes questions we hope your patients will ask as they consider going someplace for cellular therapy, and a list of what the answers might be. So these are general questions about translation. What preclinical questions need to deal with the validity of your preclinical model, not only, as I said, to predict benefit, but also to predict risk. What about species? We do a lot of this in rat. Do we need to go to a larger animal? Would it be a pig or a dog or a primate? Primates have corticospinal tracts like we do, but much harder models to work with. How do you test those combinations? And what about the idea that if I can do it, someone else should be able to do it? Because if I do the surgery, I'm not going to do all the surgeries. Things that we do in people better be repeatable. Clinically, the selection of appropriate and sensitive outcome measures I've already addressed in minor detail. The ethics and the necessity of a sham control when you're doing an extremely invasive therapy that will just getting the cells in itself will cause a number of risks, pain, and other things. So how ethical is it? When do you do it? How do you not do it? Serious question. A lot of the neuroprotective, especially the drugs that we do, are being tested immediately at the time of animals. The cells are being put in at the same time you're in the animal making the lesion. Why? Because it's more ethical to do one surgery, but we're not going to do that in people. The whole issue of informed consent with the therapy that needs to be given very quickly has already been dealt with actually in the brain injury community. And the use of clinical untested therapies, public opinion about how quickly we're going actually does influence people thinking about when something's ready to go to a trial and may actually scare people away from going to trials. The questions that are specific for cellular therapy, what are the cells doing? First off, did they survive at all? Which I think is a big question in terms of some of the work I've seen. Where did they go? Did they fuse with host cells when you're talking particularly about stem cell therapies? If the cells have an effect, what is it? We've discussed this to some extent. I won't beat it into a dead horse, but I'm going to ask you this. Go back to my Schwanzell example. Yeah, you may be able to do it with a stem cell, but is that really the best way to do it? Would you take something as dangerous as a stem cell might be when you could simply do a genetic, if you want it to just make a factor? Well, it has a lot of advantages to putting a real cell in. You may actually make it regulatable, but you better have a good reason for using a stem cell as opposed to an alternative. And it should be a reason that you can describe. What are the issues of moving to human testing? We have the proof of principle, which I already talked about, the quality of your model. This was a general slide about translation, so I've covered a lot of these. What's the source of your cells? I haven't done that yet. Purity, potency are questions I'll go into. And most of all, are they safe? Because the first trial you're going to do is a phase 1 safety trial. And what predictors do we have in any of our proof of efficacy that are really looking at safety? You have to worry about inflammation, rejection, and the more multipotential your cells are, tumor formation, and then once they're there, controlling the long-term function of those cells and the release of whatever factors they make. So I'm at NIH, I'm used to this question. You want an R01 to do what? You can't get an R01 to ask if your animal model is appropriate for a target trial or whether the outcome measure. You know, you've already done your discovery work. Optimal dosing, clinical delivery, medicinal chemistry, and pharm tox. These are FDA terms for pharm tox. But, you know, you've got a drug, you're done, like I said before, you may have to make that drug better. Doing an animal model simply to mirror what a human clinical trial might look like after you've already got some proof of efficacy data. It'd be very hard to get an R01 to do that. Controlling for other variables like rehabilitation, activity-based therapies combined with a cellular therapy can make for some pretty tricky grant proposals. And especially replication, doing the same thing somebody else already did. Try and get an R01 to do that. Try and publish that. NINDS for the last 5 years has actually had a contract specifically to do that. So we've actually been able to say, this is important, we want to do it. In fact, that contract is in recompetition right now. Interestingly, of all the things we tried, and then we always tried to pick the best one, we've never been able to replicate things exactly as they were originally published. And we've done 8 really serious trials so far. So let's get back to this pipeline. We can fund the entire pipeline, and NINDS more than the other institutes, to be honest with you. You can actually put in unsolicited grants for these and traditional exploratory grants, but at NINDS, we do have a program of translational cooperative agreements, which are specifically designed to do the boring stuff and funded appropriately. We actually have an experimental 5-year contract going on targeting spinal muscular atrophy, specifically to take it across the entire spectrum. And in 5 years, they will have gone from proof of concept to a clinical trial. Counteract is working with terrorism agents, and through the NIH roadmap, both the libraries collection and RAID, which will actually allow you to do your drug development, and other facilities, these things now do exist. I won't beat a dead horse. One of the things we really stress with our translation is, just like a drug company, we want to look at it every step of the way. Sunk costs are one thing, you can't do anything about them. But putting the next chunk of money into it is something you can do about. So these are very milestone driven, and milestones are a very important part of the review process that we put these grants under, and that's really, and it's everything. It's cells, it's prosthetic devices, it's mostly drugs, just because that's what's in development. And so far I haven't had a successful spinal cord injury proposal go through the system. They're very competitive, but I think that the spinal cord community as a whole has gotten stopped up on this question of how much preclinical efficacy you really need before you start pouring $1 million a year into something. And so review is largely an issue of getting the peers to agree. Originally, I was not the one who was asked to give this talk. My colleagues in the Cells and Biologics Division of the FDA were asked to give the talk, and we have a memorandum of understanding. We work very closely together. But anyway, they recommended to Anne that I come and do this, but the last part of this is going to be to cover the points they would have raised. So this is really what I've learned from the FDA. Leave the proof of concept behind. We're talking about a product. Products, you have to worry about safety, purity, potency, sterility when you're talking about drugs, and safety is always primary. If you want to waste your money testing something that won't work and your company will never make $1 on, that's fine with them. But don't put patients at unnecessary risk to do it. Okay, that's their concern with efficacy, because it's always a balance between efficacy and risk. The product issues. Your cell, drug, or device, or combination is a product, and that's not something most biological thinkers think about. I've got a cell, what's its potential, what's its development, this is a cool question. It's a product when you're talking to the FDA. So you can't say, well, I'm doing this in the lab, and I'm going to put that through, and when I get there, I'll know it. You have to do all the testing on the product that you're going to test. So this is a really significant issue. You want it to be labeled. What are you going to label it as? That's where the mechanism is important. It needs to be reliable, safe, and effective. You need to worry about manufacturing control, source of tissue, characterization of the preparations, each and every time you're going to put it into a person. You have to set those specifications. You have to worry about potency assays that will be reliable enough to go forward with your trial. The animal model question, everyone automatically assumes the FDA is going to force you to do 2 species. For some reasons, and in some cases, that is true under the drugs situation, but the second species is very often not the injured species. It's just farm toxin and how much does the liver get affected in another animal model. In cells and biologics, it's much more fluid because they have fewer gold standards. The questions they have is the model you're using useful for predicting systemic reactions? So they're going to talk about toxicity and efficacy and safety. And their tox trials are where you're going to get the second species if you need it. The questions I raised before, does it mimic or target the human population? They will ask that. The cells and biologics are so risky that they will ask more efficacy questions, and the behavioral model sensitivity comes up, especially to the extent that those models will predict risk. And then finally, and especially with stem cells, tumorigenicity is a huge issue. And the first thing you'll find is you'll propose a 4-month outcome, and they'll say, you really need to do 6 months, 9 months. You need to talk to them early because you don't want to do all those experiments and then have to redo them. FDA Sieber, Cells and Biologics, asked familiar questions. Delivery and dosing questions, preclinical data, delivery device. You may be using a delivery device that itself is novel. A certain way of targeting your cells and putting something onto the spinal column so you get your cells directly there may be novel enough that it needs to be regulated along with your cells. Better talk to them and find that out, because then you're talking to 2 different divisions, in which case Sieber will still be primary, but you do have to deal with the concerns of both divisions of FDA. They're very interested in cells in terms of the post-transplantation fate of those cells. They will ask you about evidence about cell migration, differentiation, and continuity and survival of the cells, the phenotypes expressed, and as well as the functional integration The more they become a permanent part of the nervous system, the longer term their effects will be, and post-cell survival. They also ask different questions than any scientist has ever been trained to ask. The top reasons, I should have had, David Letterman, top 4 reasons, but the top 4 reasons for delaying a phase 1, remember just the safety trial, is that the clinical investigators that are working with people bringing these data forward just haven't done trials and aren't qualified to do the types of things that need to be done in these trials. They send them back and say, beef up your clinical side. The investigator brochure is misleading or erroneous. A brochure? But informed consent is hugely important. Human subjects are or would be exposed to unreasonable risks. The IND, investigative new drug, which is just the document you supply, does not contain sufficient information to assess risks. Those are the top 4 reasons the FDA give for why they have to reject trials. FDA doesn't actually approve a trial, but it doesn't veto, doesn't not approve your trial. The later phases, phase 2 and phase 3, where you start really talking about efficacy, study design becomes a much bigger issue, and if it's inadequate, the trial can't go forward. Adequate science basis is not their job, they're regulators. NINDS review committees or NIH review committees worry about that more. And you have to please both if you want NIH funding for your trial. So my summary sermon is translation implies a change. You have to ask different questions, seek different types of evidence than you would in your normal everyday biology, and you end up in a different place than you began. You don't end up with your next R01. You end up handing your information off to somebody in the next phase of that pipeline. That's really the goal. You have to think very early in terms of your therapy as a product and develop it as such, and at the same time start early talking to your statisticians, talking to your trialists, and talking about what realistic clinical targets are going to be. And then aim your preclinical data as much as possible in those directions. What mechanisms do you really need to know? You really have to seriously ask yourself that question, because you have to also say, we've got a bit of Heisenberg uncertainty principle going here. Is you can, as I said before, you can drill down so deeply in the basic science that you'll never move out of that, because you'll never answer all the mechanistic questions, but these are imperfect models. How much do we really need? I don't know the answer to that. You have to develop a plan as much as possible with clear go, no go. So I was involved in a Schwann cell project to bridge. Okay, the Schwann cells alone didn't work, so you start adding other cells, and you start adding other drugs, and you start adding this, and you start adding that. Are you still talking about a realistic clinical therapy at that point? Maybe there's a no-go decision that has to happen at a certain point. You have to carefully assess your models, and please remember they are just models. They all have limitations. That's fine. If you've addressed them, that's what you need to do, and I think the reviewers will understand that. And this is about relieving the burden of disease. The hardest thing I had to do was look Augusto Adoni in the eye one day and say, should you go ahead and put some cells into a person? Is there anything else he needed to know? And he's the, I blinked just as you did that. Augusto Adoni, the Lorenzo's oil guy, had the Myelin Project was doing research, and he was ready to put the first few Schwann cells into a person, and had a group of researchers come. They'd already had their clinical design and clinical trials set up, and they had independent people who'd never been involved in it come and say, should we do this? And I finally came that day and said, you've told me everything I honestly would need to know. And it was one of the hardest things I ever did to say, I'm willing to say yes. Oh my God. This is safe enough for what they want to do. It probably won't work, but at least they're going to figure out why. And to me, that was a huge turning point in my life. Then he turned to me and said, well, Naomi, you talked to the press. I'm going to ask you another question. What am I going to get asked, and what should I say when I do? And so that was an even harder question. What would convince you? And it's hard for basic scientists to ask this question. It's a lot easier for clinicians to really say that the risk is worth the benefit. And so I'll leave you with that, and you've got more than 4 questions.